Thank you very much. I also would like to say thank you to Francesca, to everyone else. I just realized I've been coming here for 11 years, since 2012, but it's my privilege that I participated to every one of these meetings. I feel like we haven't jumped a long way as much as we would have liked to, but on the other side, we saw it from this morning's presentations and with Gécz that did we go a long way, but it's extremely challenging disorder what we try to resolve. So my talk is mostly about the theme which we initially started with, and we thought that was important because of relative ease of interventions into the hormone receptor signaling pathway. So I will continue on that. These are the people who mostly contributed to study, and I would primarily talk about the work of our PhD student Rebecca, who just submitted and will be moving on to do something else. Raman is still with us, and Tarin as well. The others, and Stefka is here, and Paul, you know as well. So this is just a reminder that kind of we're facing this challenge that when we have, and still I believe it's a kind of a good situations when we have PCDH19 constitutively, or even if we don't have it, at least in males, we haven't seen a female with constitutive loss of PCDH19 in both chromosomes, and we can consider that still as an unaffected status. And problems only happen when we see the mosaicism. So it is a disorder of mosaics, and it's very important to understand it for broader relevance also for other disorders. Now, I don't think there is any strong evidence to show that the mosaic males, post-zygotic somatic mosaic males, are identical to heterozygous females, but we attempted to do it with Christy, and we've done it using the survey, using different measures of executive function and brief and autism spectrum disorder measures, and it did look like that the mosaic males and affected heterozygous females perform very similarly. And the heterozygous unaffected females, so those who do have PCDH19 variant, but were not clinically affected on these tests, perform similarly to unaffected males, or in this case, they were to transmitting males, so the knockouts. So that to me tells what also we heard from Professor Vijayana, that it looks like that the mosaic males are pretty much identical, or as much as we can compare with the numbers what we have to the heterozygous affected females. So which in other words means that it is probably the cell mosaicism which is driving it, and not a lot more. I mean, especially the gender differences. And from the other perspective, I guess we study also, I'm not going to talk about it, this series of monozygotic clinically discordant twins, which at least 7 pairs were reported in the literature. These are genetically identical individuals, and they are highly discordant for PCDH19 clinical presentations. Not to the extreme, they are affected, unaffected, but some they are, but they are certainly differentially clinically affected. Some time ago, we looked at, of course, the hormones and the onset, and you have seen it again summarized, that the onset of seizures is usually about 8 months to 10 months, the mean, and then there is a bit of an offset. So recently, Rebecca relooked this from as many as 395 females and 22 mosaic males, and that's fairly robust. So we do see that the peak onset of seizures is within the first year of life. And we saw also that it's a predictor, as we found with Christy, of the outcome, not just in seizures, but also the cognitive and autistic comorbidities. So that's why we continue to look at this critical developmental period and what's behind it, and whether that's not a therapeutic target. So historically, we have done some expression profiling. We tapped into the pathways associated with estrogen and progesterone receptors, and we harnessed that to assess variants in PCDH19 to see whether these variants are pathogenic or not, so at least we can diagnose this disorder properly, because there are still many variants. I routinely receive about one inquiry every couple of weeks about a variant in PCDH19 from all over the world. Is this a deleterious variant or not? So it's still non-trivial. And then you know that we measured some of these neurosteroids and thanks to colleagues here in Italy in particular, Nicola Specchio, Maria Trevisano, and Professor Biagini, and these girls are allopregnanolone and pregnanolone sulfate deficient. So obviously we thought that supplementing them with these neurosteroids will help. And over a long period of time, finally, maybe some of you noticed this phase 2 placebo-controlled clinical trial of oral ganaxolone was published. The bottom line is that there was good toleration, there was great reduction in the frequency of clustering seizures compared to placebo, but this was not statistically significant because instead of 70 to 100 patients targets, there were only 21 patients recruited during COVID. And that unfortunately killed the statistical significance, but I think there is some good news in that. Of course, ganaxolone is not allopregnanolone. Ganaxolone is highly modified allopregnanolone, and currently allopregnanolone showed efficacy in other seizure disorders. And ganaxolone is also very efficacious by the look of it in CDKL5 epileptic encephalopathy, and it's been approved by FDA. So I think there's a potential perhaps to keep investigating these pathways for drug targets. So we did look at it from that perspective, that whether these nuclear hormone receptors regulate PCDH19. So we first really needed to choose a model, and in this case we went to the breast cancer cell lines, and we chose, we worked with MCF7 for some time, but we chose this T47D, which express all receptors. So we didn't need to deliver them, so they were naturally there. It's a breast cancer cell line. And Rebecca being very diligent, so she stripped the media of all hormones, so that the experiment was clean, and then she fed the cells with different concentration of estrogen, progesterone, and dihydroxytestosterone, which is the active testosterone after this period of time. And you can see that after 16 hours and ongoing, these hormones, and primarily estrogen, estradiol, but all the others have strong suppressive gene expression effect on PCDH19. So they suppress PCDH19 expression. And that was fairly robust. We did some studies, and we tried to generate more data in male and female cortical neurons for E14.5, and we saw a similar effect primarily for estradiol. And interestingly, statistically significant only in female neurons, but not necessarily always in male neurons, even though the trend was similar, which suggested that estradiol suppress PCDH19 expression also in neurons and not only in breast cancer cells. We tried to check whether, of course, based on our gene expression studies, that this regulation is driven by estrogen receptor alpha, which was the obvious culprit because that's estradiol binds to. And we saw after siRNA downregulation a derepression of this activity, which suggested strongly that this repression of PCDH19 by at least estradiol is driven by estrogen receptor, and we didn't see such a strong response with respect to the scrambled with siRNA against progesterone or androgen receptors. So the estrogen receptor alpha derepression, knockdown derepresses PCDH19 expression. So, which means that PCDH19 is at least to some extent regulated by estrogen receptor alpha. Now, we didn't find any estrogen receptor binding sites in the promoter of PCDH19, so we had to look at other transcription factor which probably helps estrogen receptor to regulate PCDH19 expression. We found FOXA1. FOXA1 is a bit of a beast. It's one of the major pioneer transcription factors which opens the chromatin and primarily at enhancer sites. And we found two sites at this position from the transcription start cell of PCDH19, and we targeted FOXA1 with siRNA, and we de-repressed again the expression after treatment with estradiol, suggesting that FOXA1 is involved in recruiting estrogen receptor to the promoter of PCDH19 as the pioneer transcription factor. And then, of course, Rebecca took some of these binding sites and mutated them. Here you say the vehicle, here you see the wild type, and what's when the sites are mutated in the reporter, it's de-repressed, which again was another independent evidence that likely these sites are functional, and it is FOXA1 which recruits estrogen receptor alpha to regulate PCDH19 expression, at least in T47D cells. So generally, FOXA1 is an important pioneer transcription factor which recruits steroid hormone receptors to different genes to regulate their expression during development. It has not been studied in neurons in the brain. It's mostly in prostate cancer and breast cancer where you see most of these studies. There is more than 1,500 publications about FOXA1. It's quite amazing. Now, interestingly, relating to work of Maria Passafaro here and her discovery of the link between PCDH19 and, for example, LSD1, an immediate early gene expression regulation. Now, FOXA1 is in that neighborhood where LSD1 has dual function and major regulator of androgen receptor transcriptional activity, and FOXA1 is a part of that regulatory complex. So then there was obvious that the question, does PCDH19 interact directly with estrogen receptor? But we didn't see it, or progesterone receptor. We didn't see any interaction of estrogen receptor and progesterone receptor with PCDH19. However, when we looked at interaction with androgen receptor, we did see quite clear interaction with androgen receptor and also cleavage, which had been described before when androgen receptor is cleaved, which I don't know specifically what the biological significance in this context is. So this is done in HEC293 T cells, again in a relatively artificial model system, but it looks like PCDH19 quite confidently interacts with androgen receptor. And all I can tell you, this is not only a property of PCDH19, but also other non-clustered protocoadherents. So we believe it's robust, and it's not only PCDH19 specific. Now, then we try to convince ourselves that we are on the right money from a very different perspective. So we have collected over 20 different cell lines from affected PCDH19 females, these primary skin cells, transmitting males and control males and females. These were passage and age matched where we could, and perform RNA sequencing as well as methylation array studies. Now, from the methylation perspective, it was interesting that based on the significantly differentially methylated regions, the individuals in this experiment clustered pretty nicely. The pale blue are male controls. These are transmitting males, and these are female controls, and the green are affecting males. So they nicely cluster based on the PCDH19 genotype. And when you look at the significantly on gene ontology enrichment of transcription factor targets of these promoter regions, which are the differentially methylated regions between affected females and female controls, you see enrichments of target of androgen receptor, you see enrichments of beta-catenin, and you also see zinc 217, which we know it's a LSD1 interactor and works with LSD1, which again suggested that there is a link. What is intriguing in this, that this is from skin cells. So this is an epigenetic imprint, which probably is, these marks are put on this DNA fairly early in development, because we're finding in the skin cells. So what this means that we found this dysregulation of nuclear hormone receptors, but we also found masculinization of gene expression, which was rather intriguing. So we look for estrogen receptor, of course, and androgen receptor, and especially androgen receptor is significantly upregulated. As you see, like perhaps with Gécz, nice example of the thermogenic seizures, that there is a spread that not all heterozygous females would have very high androgen receptor expression, but some do. But generally, on average, it's significantly upregulated. This hierarchical clustering of the differentially expressed genes, so this is a transcript level, is also interesting, where we see the male controls sitting separately to female controls, transmitting males sitting closer to the affected females. So from transcription perspective, the transmitting males actually look more like affected females or control females rather than control males. So it is not just masculinization of gene expression in affected females, but it's feminization or the opposite effect in the transmitting males, which is kind of interesting. So when you look at, again, the graphs which I told you, developmental timing of PCDH19 expression, to summarize it, and we look at some of the published work here from people in the room, in animal models, we see that peak of expression early postnatally or after, and that's probably the timing also of that mini puberty and expression of hormones. But technically, what I guess our work in summary in a nutshell suggests that if you want to decrease PCDH19 expression, probably hormonal treatment with E2, P4 or dihydroxytestosterone can achieve it as much as antisense oligonucleotides, potentially. Whether that's a simplified statement, it could be, because of course hormones do other things, but we believe that probably could be explored how these hormones can suppress, and maybe that's the benefit in puberty with the suppression of the seizures, at least some of these individuals, or I probably could say majority, also maybe not all of them are old enough to tell, suppression of the seizures because of the surge here in steroid hormones, which technically suppress PCDH19, which brings these individuals more to the null level and as such unaffected status or less affected. So looking at this picture, and we have seen it so many times, when I was thinking about it, that, well, we know this is wild type, we know this is a loss, and this is unaffected, but different genotypes. And then we have these affected, and I think even Crystal Depienne in her first paper, she put it like this, but I think we need to think about this, that it's not probably two, or it can be both, that it's these separate networks, but it's also these mixed networks. And I'm kind of curious more to look at these connections between the PCDH19 and PCDH19 positive and negative neurons and how they drive the disorder, because that looks like where the things are happening, also based off what Maria and her team and Sylvia and others were showing. So from that perspective, we now know a lot about PCDH19, what it's doing over time. I was just picking up in Hishashi Memori's paper from Science a couple of years ago, where they found in hippocampal neurons that there is different beta-catenin clustering at the pre-synapse, so at the PCDH19 positive side. So in this case, the post-synapse was PCDH19 negative, and the pre-synapse PCDH19 positive hippocampal neurons. There was a different or reduced clustering of beta-catenin. I was intrigued by beta-catenin for many years, since pretty much the neuronal Rosette's findings. So we have done PCDH19 proteomics and 2 different experiments, thanks to also Stefka and Paul providing us with reagents. And we identified many proteins in embryonic stages, about 28, and postnatal about 32, of which about 14 proteins were common to these different stages and fairly robust with respect to these different controls. And these are the gene ontology enrichment of the proteins. So far, when looking at the literature, we found across 13 studies 238 unique PCDH19 interacting proteins. I mean, this is massive. How much of this is really part of the complex? How much of it is direct interaction? How much of it is meaningful? How much of it passengers and need to be validated? It's hard to tell, but we definitely validated PCDH19 interacting with beta-catenin, and in view of the Memori's science paper and beta-catenin clustering at the pre-synapse at the PCDH19 positive sites, and the study Maria and her team published about the cytoplasmic domain, we thought it was very interesting. So Rebecca did go back and validated beta-catenin interaction by co-IP with PCDH19. We also tried different fractions or parts of PCDH19, and we tried mutations, but that really didn't affect the interaction with beta-catenin. So PCDH19 interacts with beta-catenin, we believe, and in mouse brain, not just in vitro. What was really intriguing when we tried to do the top FOP flash assays, and when we Wnt activated provided ligand Wnt3A, we saw a huge hyperactivation using the cytoplasmic domain, which would suggest that on the side where there is PCDH19, there is probably improper cleavage because of different interactions with N-cadherin on the other side, because we know the N-cadherin overexpression can rescue that, and that might somehow hyperactivate Wnt beta-catenin signaling at the PCDH19 positive sides of the neuron. And whether that's a part of the story, well, we probably don't know 100% yet, but PCDH19 promotes, we believe, Wnt beta-catenin transcriptional activity in vitro. Now, again, when we went back to our cells of our patients, skin fibroblasts, we saw many different targets of Wnt beta-catenin signaling pathways significantly dysregulated. And again, when we clustered our different genotypes based on these genes, we saw that these are female controls, these are male controls, they clustered together, but now suddenly affected females and transmitting males, so the knockout males clustered together. So as you would expect by the genotype to have altered Wnt beta-catenin signaling. And Frizzled1 was one of the genes which is obviously affected. Androgen receptor, if you note, it's another target of these pathways, which again would make sense that, as you can see here, it's significantly upregulated in the genotypes which are mosaic or a knockout for PCDH19, while in controls the levels are lower. So in summary, we believe PCDH19 regulates nuclear hormone receptor and androgen receptor in particular, as we found it from patient cells. Hormones regulate PCDH19, so there is a loop how these molecules interact with each other. FOXA1 is an important co-regulator and recruiter of estrogen receptor to PCDH19. What we believe is that this axis of PCDH19 nuclear hormone receptors is very important early in development, because you can see it in skin cells. So it's obviously must be happening early. How important it is or not in what we do postnatally, like we don't see anything until the first seizure, whether that's there are some other events happening, or maybe it's not so severe and everything pretty much starts after the mini puberty at the age about 6 to 8 to 12 months of age. It's yet to be determined, but it looks like that we should be able to recognize PCDH19 epilepsy based on epigenetics. So based on early developmental events which leave mark on DNA, epigenetically and transcriptionally. There's a lot of interactors, PCDH19 interact with androgen, beta-catenin, and it looks like that when beta-catenin signaling is dysregulated significantly in patient cells, and these two pathways as such are good therapeutic targets, also for the purposes, also for the reason that there are different drugs available. Also, we don't know exactly how many of these are FDA approved and could be applied for the patient benefit at this stage. And I guess I'll leave you with the people who did most of these works. Thank you.
Questions for Gécz?
5 minutes.
For Jozef.
So beautiful work as always, Jozef. So how are you thinking about, it seems like effects that you see in both the transmitting male and the heterozygous female aren't disease phenotype related. So yeah, how are you?
Yeah, that's a very good question, and it really bugs me and bugged me for a long time. So that's why we have done these transmitting males, even though they are unaffected, and we collected the material, which was not always simple, to tell us that there is a transcriptional and epigenetic impact, but there must be something else happening, which is probably the cellular mosaicism, which is another factor which tips the system over. And as such, we need to look at it that we probably will be finding functional and maybe developmental differences, which actually might be physiological in a way that they are not necessarily disease relevant because the transmitting males would have them. But they are actually helping us to separate the important events by comparing them to affected females. And so we're using them as another level of control where we see how the biological system, how robust it is, there are a lot of compensatory pathways, and it looks like in this case, in the PCDH19 knockout males, there's a very strong compensation. Now, what is the compensatory mechanism? It's the question. I can give you only a hint because we worked for different reasons with beta-catenin, and we have generated knockout and heterozygous knockout beta-catenin cells. And most frequently dysregulated and common genes during in vitro neuronal differentiation of these CRISPR-edited human ESC cells, male ESC cells, is PCDH1917. So it is the pathways, which is probably quite robust. We know it from the PCDH19 different interactions that they do interact together. And maybe at the stage of the rosettes early differentiation, maybe it's a 17, which is there and maybe can take a lot of roles. Maybe even 19, it's important slightly later. But there is clearly some redundancy for unknown reasons during evolution. And we can live without PCDH19, but we can't live halfway without it, kind of. Now, why? Whether it's a beta-actin activation or whether it's a response pretty much can collide on the immediate early genes, as Maria found through the C-terminal domain of PCDH19. It remains a question whether that's a driver, but I feel like we should look at those connections of the positive and negative neurons, which are probably driving the phenotypes in different types of cells with different clinical outcomes. Isabel.
So when I read the science paper with hippocampal phenotype, I actually didn't think of brain signaling as an issue, as dual roles. It has the signaling role, but it also plays a very important role connecting catenin to the actin cytoskeleton. So, do you know what is known about Wnt signaling in adult neurons? Is it relevant for the neurons? Is it something that is going on? Is it something that is altered by the levels of catenin expression?
I don't know. We know a lot more about canonical Wnt signaling more than non-canonical, and again by studying because beta-catenin is we look at beta-catenin as an important gene in cerebral palsy, kind of movement control. So we studied these cells, but a lot of we studied certain missense changes, which not necessarily tap into canonical Wnt signaling pathways. The question is how is the non-canonical Wnt signaling pathway plays a role. What I found interesting and the importance of the N-cadherin that they show that overexpression of N-cadherin rescues that, which was quite amazing. Which again, I talked to Hisashi. He didn't think necessarily that was beta-catenin mediated, but he admitted that they haven't looked. So the question is, I mean, even if your model, Jack, whether you look whether it's hyperactivation of beta-catenin, because that's all adherence junctions, that's all kind of neuronal rosettes and related processes, and whether that has relevance, I don't know. So we probably still a lot more focused on the canonical signaling pathways, and there might be other roles.
So, unrelated question. As you know, we're looking at one of the genes we're looking at. VUS interrogation is PCDH19. So, what is your most robust kind of cell culture test to show loss of function?
Good man. I don't think we have a robust one. To be honest, we still have a couple of variants which we run through the estrogen receptor reporter assays, and they were pretty good. Now we're trying to repeat them through the androgen receptor reporter assays, given the effect on androgen receptor. We are not complete that. We haven't completed. Haven't done. Like you mean binding in terms of?
Looking at interaction.
These missense changes, well, estrogen receptor doesn't interact with PCDH19, which we thought it would, but doesn't. And I don't think it missense changes affect interaction with androgen receptor, because the interaction is probably happening in nucleus. Yeah. So we don't have a robust assay for the missense changes. Yeah. But we have a few interesting missense changes in the cytoplasmic domain, which we still throwing our hands up because we don't know what they do. But there are some very interesting, very specific missense changes with very specific phenotypes in males. But that's all. The MutePred seems to be performing the best in silico prediction tool for PCDH19 variants. That's still our best tool. Provian as well, partly, but MutePred. Otherwise, we don't do them as much. It's just probably too much work, and I still don't think it provides a very robust answer. Yeah, that's probably it. Lunch?
Lunch.